The problem that I want to speak about today is shelf life. We talked about traits that consumers care about, and to get genome editing accepted broadly, several things have to happen, in my opinion. One is that we have to have traits that consumers care about, not input traits. And secondly, the people doing the talking probably need to be university professors and small companies, because I think in general those populations tend to have more credibility with the public than the big ag chemical companies. So consumers certainly want very convenient. They want convenience. They want long shelf life. It's unfortunate when you buy bananas that they go rotten, or you buy roses and they go bad very quickly. It turns out it's a huge aspect of food security, in that we lose about a third of our crops every year. The economic value of that is almost incalculable. I can't get my hands around $1 trillion of loss, about 70% in the developed countries and about 30% in the developing ones. There are several conventional solutions, at least 5 different physical or chemical methods. I don't have time to go into these, but what I want to emphasize is that our genetic technology is completely compatible with all of these physical and chemical methods. And by combining 2 or 3 of these along with our solution, it's possible we might be able to get vegetables to last 50 days with no loss of quality and no pathogen issues. So the big idea behind the founding of our company was that our customers are innovative seed companies or processors. They're not farmers or consumers directly, and they're dissatisfied with current attempts to improve shelf life. We have patented technology using genome editing that increases shelf life by 2 or 3-fold. In addition, the same trait increases the yield and abiotic stress tolerance and provides a little bit of pathogen tolerance. We do this by making a small deletion in a critical gene involved in cell fate that's present essentially in all plants and all animals and even archaea. And this is not a science project. It's not just in the lab in the greenhouse. We have validation in field trials. And by doing it by genome editing, after we validated the target using transgenic approaches, we're now going to work through our partners to commercialize it through genome editing, and this avoids the GMO controversies. It's kind of embarrassing as a PhD in genetics from Stanford to talk about my science in one slide, and it's a cartoon. But I think this will illustrate the point. There are 2 genes involved in this switch, EIF5A, which stands for eukaryotic initiation factor 5A, and DHS, which stands for deoxyhypocene synthase. The product of the DHS gene post-translationally modifies the EIF5A to form a hypocinated form. This is a unique PTM. It's essentially a spermidine hooked onto a conserved lysine. It's the only, EIF5A is the only protein in the cell that's hypocinated, and every cell is always monitoring the ratio of the undecorated form to the decorated form. When there's a predominance of decoration, the cell goes into apoptosis or senescence and dies. And when there's a predominance of the unmodified form, the cell continues to grow and divide. My co-founder, John Thompson from University of Waterloo, discovered about 15 years ago in senescing leaves of Arabidopsis that the level of DHS went up about 15-fold. So doing what any good biotechnologist would do, if you see something nature does that you don't like, do the opposite. So he downregulated DHS using antisense, and he found out whether you reduce the amount of DHS and therefore hypocination, or alternatively, if you increase the amount of the target of that EI5A, in either case, you would, as a result of flipping the switch in favor of continued growth and development and away from cell death, he found that these plants not only had longer-lived leaves, but also perishable products in multiple species, increased biomass and seed yield, and enhanced tolerance to stress and disease. This is all by inhibiting this fundamental switch common to all plants. He showed this in annuals and perennials and trees and in woody crops and vines. Every crop that's been tried to some extent has been improved. Science is always more complicated than a simple cartoon. It turns out that EIF5A comes in 3 flavors. These are expressed differentially in different times and tissues. However, if he downregulated DHS, he got positive effects with all 3 traits. Here's some evidence that's buried on the poster here with tomatoes and bananas. Again, antisense downregulating EIF5A1 or DHS. The tomatoes lasted two or three times longer. Same thing with bananas. This top row reminds me of bananas that I get from Costco. They're either underripe or over the hill. And by downregulating DHS, the bananas last about twice as long. Same thing with carnations, about threefold longer shelf life. My mentor, Jerry Calder, told me that this is very interesting, that shelf life of carnations is enhanced, but it doesn't really mean much because carnations last long enough already. But if you can do this with roses, that would be a real killer. And in bananas, field trials were done by our partner, Rahan Meristem in Israel. And you can see black sigatoka trees are infected in the leaves, and using downregulation of the pathogen form, they seem pretty healthy. These were interesting results, but not commercially good enough. So they viewed this as tolerant but not resistant, and they're waiting until another gene can be stacked with this before commercializing it. In the greenhouse, we saw similar results with Pseudomonas syringae infection of tomatoes, tolerance but not full resistance. The same thing in Arabidopsis. The yields went up between 50% and threefold. And this translated to the growing plants as well. It's really obvious. Even in alfalfa, when this was done, you can see T0 plants being obviously healthier, growing faster, greener than the controls. And in canola, you could see not only the leaves bigger, they're also photosynthetically more efficient, and the root architecture is much greater. And we think this underlies some of the drought tolerance and enhanced nutrient uptake. It doesn't change the nutritional composition. This is very upstream. It's not like ethylene or hormones or other downstream PPO. It doesn't change the nutritional value. The fatty acid compositions are pretty much identical. And it looks like this gene is expressed in the roots, which is consistent with DHS, where also that's a root preferred gene. And going down even lower, it looks like it's in the companion cells around the phloem is where the growth isoform of the IA5A is most expressed. I mentioned in alfalfa field trial, this is the reason I started the company, because I understand my customers, seed companies, typically require field trials in order to validate a crop. Otherwise, it's just a science project. And over 2 years, in an elite line of alfalfa, the yields went up 20 to 45% in an elite line. That's unprecedented. Alfalfa is a crop, very important crop, that has not really had any yield increase in almost 20 years. And this field trial showed a statistically meaningful increase. I showed this slide very proudly at an angel group meeting once, and afterwards a woman came up to me and said, "Jerry, that's the worst slide I've ever seen in my life." I asked her for help. She said, "I can't help you." So I did it on the plane, and I narrowed all this stuff down. I picked 3 transgenics versus the control. First year with 2 promoters, second year with 2 promoters. Narrowed it down to here. Green is good, yellow is okay, and red is good. So this is the way things are. This is the way things are classified. But you can see over 2 years the colors are pretty much similar between the upregulated ones and the controls. And here's David Johnson sort of decapitated with a wild-type plant and an upregulated growth isoform of the IA5A. The important thing is the yields go way up, and the quality is pretty much the same. We have a lot of patents, so please don't infringe. I don't have time to go into the business model, but we're partnering with other companies to get this done. We're both doing licenses and co-development. We have a very experienced team. The total combined experience of the core team of 5 and our 6 consultants, I think, is something like 250 years. I'm not sure that's a good thing. We've got a lot of traction. We've got licenses in license from Benson Hill CRISPR 3.0. That was announced about a month ago. We've got licenses or co-development in alfalfa, in coffee, tomato, potatoes, and in not to be named peanut company. So in summary, we'd love you to join with us to build value. We have proven technology. We've got a very experienced team and very good traction. I want to conclude with sort of a personal note. People go into startup companies for a whole lot of reasons, but I just want to assure you that I've been in this for a long time. I've defined 3 different emotional states of an entrepreneur. This is me when I was 10 months old and I had just closed a deal. This is the best time to be an entrepreneur, when you're raising money or closing a deal. But you have to spend time figuring out your cash position, how you used your funds, and how much you have left in the bank. So I was busy counting on my fingers at that time. It was before Excel spreadsheets were available. And finally, anxiety, which is all the other times. So I'd like to thank you for your attention and open it to